Hello, really excited to introduce Nikos to you today. Nikos joined the Center for Alzheimer's and Neurodegenerative Diseases last year, and we were really lucky to recruit him from a terrific group in Belgium. Nikos got his undergraduate and PhD degrees at the University of Athens, and then came to KU Leuven to work in the Switch Lab, which is run by Jos Simkowitz and Fred Rousseau, studying amyloids in biology. And Nikos has had a long interest in this. And Nikos's work is really innovative and groundbreaking because he's developed both computational algorithms to predict amyloidogenic sequences and propensity to form into disease-causing amyloids, but also to predict these sequences throughout the proteome and thereby also make hypotheses about cross interactions based on sort of co-aggregation of proteins. And this is an area of great interest to us in neurodegenerative diseases, which are so often associated with amyloid formation. So he's bringing this combination of computational biology and rigorous biophysical and biochemical approaches to study the biology of amyloids in a variety of neurodegenerative diseases and systemic amyloidosis as well. And welcome, Nikos.
Okay, good afternoon, everyone. First of all, thanks to Mark for that very kind introduction, and thank you to the committee for organizing this forum. It's been an interesting chance and opportunity to see cool stuff that are coming at UT Southwestern. In the next few minutes, I'm going to be discussing about some of the research that I've mostly done during my PhD and postdoc years, and how we are trying to use that as a foundation right now to develop some potential solutions in protein aggregation related disorders here in our lab in the Cairns. So I always like to start my presentations with the simple question of why. Why would we care about protein aggregation? And in my case, it's quite an obvious response. So that's another reason why I like to start it that way. Protein aggregation, or more specifically, the formation of beta-rich fibrillar aggregates known as amyloid fibrils, are associated to a huge number of widespread debilitating diseases, including major neurodegenerative disorders such as Alzheimer's disease, Parkinson's disease, prionopathies, and so on. But also other forms in the rest of the body, like type 2 diabetes, for instance, systemic amyloidosis forms, and even certain forms of cancer. So you can hear the variety of these different types of diseases. Naturally, if you group them together, they're currently the second leading cause of deaths worldwide. And the projections are actually much more dire because based on the World Health Organization and with the expectancy of life and quality of life increasing over the years, we're expecting in the next few decades that they're going to take over and be pretty much the main reason of death worldwide. They're also associated to a huge economic burden, both in our healthcare system, but also in terms of caregivers and families that are suffering from these diseases. Yet, despite this huge impact, we have achieved very, very little in terms of potential therapeutics for this entire ensemble of diseases. One of the reasons for this could be the complexity. Different proteins, different tissues, different targets, and so on. So where do we start? Actually, early on, amyloid diseases were following a central dogma, kind of, in this field, where people thought that a different protein would misfold, aggregate, form these ordered aggregates called amyloid fibrils that look similar in all of the cases, despite being formed by a different protein, and then lead to a certain form of disease. So this was the general dogma initially. However, in recent years, we now have some structural information about how these amyloid fibrils look like. And here you can see a quick group of cryo-EM structures for different amyloid fibrils from different proteins. Now, although some of that previous sentence was true, like the fact that they are beta-rich and you have basically a monomer stacking along the axis of a fibril in each case. The actual structures, as you can see, do not resemble each other at all, depending on the different protein. And actually, this complexity increases even further, considering that single proteins, such as tau in this case, or alpha-synuclein in this case, have the ability to form different structures depending on the disease that they're associated. So a single protein can be associated with a number of different diseases and form different amyloid strains or polymorphs in those diseases. And to make things, of course, even worse, we also know now that it's very hard to study these, at least in the lab, because in many cases, when we use recombinant material or synthetic material to try and recapitulate their aggregation propensity, we end up with amyloid fibrils, at least at the level of structures that look nothing alike the pathological forms derived from patients. So this actually makes it much more complicated, at least in terms of diagnostic development. So some of the key questions over the years were to try and decipher molecular determinants that drive the assembly of proteins. A lot of focus has been done in extrinsic factors like the disease context, the different cells where these aggregates are formed, for instance. But some of the work also focuses on the proteins themselves. Are there specific intrinsic factors in these proteins that actually drive their self-assembly? And this was actually a main focus of my studies. This actually focuses on what is known as aggregation motifs or aggregation-prone regions. All of these terms have been used over the years, which are essentially short sequence segments that are usually found buried within the native folds of proteins. They're actually super hydrophobic and have a beta propensity, therefore stabilize typical native folds of proteins. Under given conditions, these segments are believed to potentially be exposed. So when a protein is misfolded or not properly folded in a cell, they can get exposed, and then they can self-assemble with themselves and drive the entire protein towards aggregation. So this was actually a simplistic model that was believed to be the process of nucleation over the years for proteins. This is actually a shameful advertisement in case there are some graduate students that are interested in joining the lab. But the actual point is to show that a lot of studies have focused over the year in highlighting aggregation motifs in different proteins associated both in pathology but also with functional amyloid states of different proteins. These are just examples that I did during my PhD, but there's several other groups that have worked on identifying these kind of motifs in proteins. However, the key point from the previous slide is to take that we've established that these motifs have an important role in nucleating different proteins, but we found out that they also have different functionalities at the same time. One of which came through analysis, thermodynamic analysis of the stability of different amyloid fibrils. So we now had all of those beautiful structures that I showed you at the previous slide. We developed some computational models to try and identify which specific regions of these fibrils stabilize them. Is it the entire sequence? Are there different parts of the sequence that stabilize them? And interestingly enough, we found that for different polymers formed by the same protein, despite the fact that they could be completely structural variable, so have completely different structures in the three-dimensional space. When we look at the thermodynamics, and I'm just showing some examples here for tau, A beta, and alpha-synuclein, but we did this for other proteins too, a common pattern emerged. So we could see that there were specific segments highlighted here in the shaded regions in all cases that seemed to stabilize these different structures. So there were short segments within the cores of the fibrils that were consistently stabilizing them, whereas the rest of the sequence was usually destabilizing, or simply put, not happy to be in the amyloid conformation. And it just so happened to be that these segments coincided with what we previously knew as nucleators of these proteins, so the aggregation motifs. And then through cryo-EM analysis, I'm just showing one example here, we could find that certain amyloid motifs can also reproduce the structural variability of the full-length protein when studied in isolation, suggesting that its potential structural variability could also promote the disease-related polymorphism of full-length proteins. Finally, we also did some cellular studies where we tried to delete some of these motifs, induce the aggregation with exogenously derived aggregates from patient samples, and see if this can affect the seeding potential in a cell aspect. And we could see that depending on the type of motif that you could delete from a given protein, in this case it's for tau as an example, we could see that you get differential effects, differential seeding efficiency. So there are specific motifs that if you delete them, you can completely kill the seeding efficiency, meaning that these are central for the nucleation of the protein. Others can simply reduce it, like in this case, and others can even improve it, suggesting that they might not be needed, at least for this specific polymorph. And the interesting thing was that if we mathematically try to analyze this data with principal component analysis, we can even decipher the source of the aggregates that we use for the seeding potential, suggesting that these kind of constructs focused solely on aggregation motifs could have a potential diagnostic potential. The most important property, however, of amyloid fibrils and why they are extensively linked to disease is the fact that they are extremely sticky. They can stick to a lot of stuff. So they stick to a lot of different biomolecules. They've known to stick on membranes, on small molecules, on different types of proteins, and so on. And this stickiness can lead to disease because it has been shown that different interactions with different proteins can modulate the structure of the fibrils that are formed at the same time by a given protein. But also the amyloids themselves can sequester important factors, biomolecules in cells, leading to potential vulnerability of those cells, toxic effects, in other words. In a series of studies, and I don't have time to go in depth through this, we used a hybrid approach, and we actually showed that the proteomic content of different cell types, at least in the brain, is actually susceptible to co-aggregate with key amyloids related to diseases, such as in Alzheimer's, for instance. And this vulnerability is actually defined by homologous motifs that proteins can actually harbor that have a high sequence similarity to known motifs of amyloid proteins. In other words, the same motifs can actually act as hotspots for co-deposition of other proteins, therefore leading to their sequestration. And we were able to show that this can actually be linked to selective vulnerability seen in different neurodegenerative diseases, but can also promote the co-deposition of different amyloid proteins, leading to certain forms of disease, such as cerebral amyloid angiopathy. Mark mentioned that we were also heavily invested on computational analysis of these motifs, since their importance was clearly shown. So I've developed a few tools over the years. First of all, since I've analyzed more than 2,000 of these motifs, I think experimentally at this point, I decided that it would be very useful to have a database with experimental data for proteins and their motifs that's publicly available. I also developed, using machine learning, a predictor for aggregation motifs from sequences. So if you guys have a sequence that you care about, you could easily go online, throw your protein sequence in there, and find out if you have specific motifs that could make your protein vulnerable. I must point out that this tool actually was able to identify aggregation motifs of different types of propensities rather than just hydrophobicity and beta propensity. And we were able to find that actually a lot of proteins have exposed motifs on their surface, meaning that they don't necessarily need to misfold in order to aggregate. We developed a database for the structural and thermodynamic classification of amyloid fibrils, so you can find all of that information there. And some of the recent work, we developed a thermodynamic profiler for co-aggregation propensity. So in this case, we developed a very simple model where we do some thermodynamic calculations at the growing ends of amyloid fibrils, the reactive ends, and we can actually determine whether a given sequence has the propensity to co-deposit with a given amyloid protein, or we can also use the same tool to try and develop designs that actually bind to the surface of the aggregates and block further incorporation. And by using this approach in the last years of my postdoc, I was able to turn the table into the stickiness of amyloids, use the stickiness of amyloids in a therapeutic aspect. So we used our tool to try and design peptide sequences that would block aggregation. We did this for tau. And here I'm just showing some experiments in PS19 mice where with the addition of the inhibitor that we developed, we can actually almost completely delete the formation of aggregates in PS19 mice. We did this by targeting an amyloid scaffold that leads to the formation of bacterial biofilms, which gives them resistance. And we were able to show that our peptides have the ability to completely break down the formation of biofilms. And we also took another approach where we actually tried to induce further the aggregation of a given target. And in this case, we targeted pathogens, so like viruses, bacteria, or even cancer cells. We picked up a protein that's specific to those cells, but not to the host, find the aggregation-prone regions, make a bomb, a peptide bomb, essentially, that will induce the aggregation and inactivation of that protein, kill the protein, kill the pathogen. So we actually were able to show that. I'm finished. A quick slide about our future plans in the lab. We're a new lab. We just started a few months ago. We're trying to move at the same direction. So we're doing a hybrid approach. Again, in this case, we are now trying to do a lot of protein design, AI-based protein design to develop potential inhibitors for these aggregation-prone proteins. We're also trying to develop some more customized designs in our lab, such as peptide microcycles and so on. We're trying to use the same models to decipher the limits, the sequence structure limitation of different amyloid strains that I showed earlier. And of course, we're still looking into the effects of amyloid motifs in different types of diseases. And very, very key, we are using a hybrid approach to try and do this in our lab. So a lot of people to thank over the years, so many collaborators and so many different projects, specifically the people in my previous lab where I came from in Belgium, and of course a lot of the people that have welcomed me in the Center for Alzheimer's here in UT Southwestern, a lot of which we are collaborating with already in some of these projects. And finally, we now have a nice team core that's assembled in the lab, but we're still hiring at all levels. So if there's people interested in joining and doing some of the stuff that you just saw, we'd be happy to consider. Thank you so much.